But also, it's a wonderful of you to say such kind things. I don't know if I can live up to what you've said, but what I can say is that being here has been a delight for me, simply because I feel as if I'm in a quantitative space and I'm wrapped around by people who understand things begin with the letter Q, and it's really good. Today's talks have been great, and those of you that I've met, I've really enjoyed meeting, and I do hope we can continue to have these stimulating discussions that I've enjoyed so much. So today what I really want to get across is just how complex it is to do what we're trying to do, and just how, I think you saw some input of that, how challenging it is to move from data that you'll finally manage to get hold of and interpret to a point at which you can impact that, and as they say in America, action it. Okay, so actionable interpretation of data. And because genomics is so high dimensional, or omics is so high dimensional, it's an obvious place to go there and look at this concept of action. Just as any conflicts of interest, I'm a consultant to a company called Biogen, and I've stolen heavily from a colleague of mine, Ross Altman at Stanford, who every year publishes something called Top Translational Informatics. If ever you can get hold of that, it's a set of 10 papers that he decides are the most impactful and interesting. They're usually awesome, and his choice is very competitive. It's to get on his top 10 list is an amazing thing. And what's exciting about that is that I'm just stealing some of his ideas based on that to guide us through our discussion today. So first of all, thank you, Neil, for your introduction to translational medicine, and I think you're spot on. There really is a need to do that health improvement, and you're working with people who work for you, and some of whom who die. So it's really in your face. And as a result of that, I became quite interested in saying, philosophically, we are very blessed with this ability to interpret data, and we have a responsibility to action that. And the mantra of that, the one kind of off-topic message I want to give to this audience, because it's a teaching school, is that I've been very fortunate in working with Oliver in a biostatistics department at Harvard, where we have some of the best students in the world, and they will wax eloquently about a transform that they've done or some other aspect of some elegant design, but they've got absolutely no idea how that will be useful to anybody. And if they do have an idea, they can't action it, and they don't really care. And unfortunately, that is a propagation that I think we have to think very careful about in today's world, because effectively we have a responsibility, if we're going to do anything to do with health, to make sure that what we do is actionable. So interpreting that in terms of a timeline, if we take a look at the concept of translation, in the 50s we were looking at biochemistry, physiology, as Sidney Brenner calls physiology, systems biology. Moving forward, we had affinity assays that would allow us to do things like measure the affinity of a particular substrate. And then basically the first of the hits, which was university science, started to talk to industry. And then in the 80s, the big hit, just prior to the sort of double hit that came with molecular biology, was monoclonal antibodies. And these allowed fishing out and attacking particular events in the biology of the system. And now, in the 2015s, we're looking at drug budgets, which are basically driving the ability to not fail. And that ability to not fail is really high, because most drug studies fail because they don't have enough comprehension of the system that they are addressing. And we have to get to the point at which they do. And so the concept of preclinical validation is coming up to a fore, and I'll mention that in a while. So the picture, most of which I tried to draw, I wasn't stoned when I did this, but I felt like it afterwards. Somebody else did this once. I don't know who it was, some Japanese guy. So, sorry. No, I'm very influenced in my life by the wave, and actually it's something I really care about. The whole genome sequencing arena is just the beginning, and actually whole genome sequencing really hasn't begun. It's just a very early technology. And it comes from this concept of a genome project, and the Human Genome Project, as you know, which has a huge amount of effort put into doing one comprehensive genome that was actually a conglomeration of a number of people, then has driven now the analysis, or the post-genomic analysis, which, let's face it, is association-based. And we're really not looking at causal basis, we're looking at association events, and there have been functional interpretations of the genome. The ENCODE project is such an example. And now we're getting to this awesome idea, the idea of a quantified patient that allows us to think about being predictive or to be predictive. And some evidence of that starting to occur is quite exciting. And in the background is the buzzword view of big data and how we handle this particular issue. So Lee Hood, who is a technologist that basically drove the development of the automated sequencer in the very late 80s, is basically the person who is now at the Institute of Systems Biology, but in the 90s, described biology as a data discipline. He's the first person that really started to push that concept. And actually, SmithKline quickly understood that. George Post was running the company at that time, and they dived in and went to a bioinformatics meeting and asked individuals at that meeting that were slightly interested in data biology, in computational biology, to come and talk to them in a particular room. And they hired nearly a quarter of the field in the United States. And you knew who'd been hired because they'd come out with little toothbrushes and toothpaste packets in their top left pockets, which was the freebie they got for the interview. Some of them still work for the company. Along with that paradigm of moving biology into a data science, biomedical science, is that clinicians are now moving from the as you described it earlier, self-encrypted handwritten record to the ability to type that information. And the minute it's digitized, that ability for it to fly away is there. And in terms of anonymity, we had a good discussion about that. But basically, the electronic health record, which is a novel concept which is finally coming to ground here in the UK, means that you can move from the concept of hospital IT to innovation and industry. And the concept now is how we address that, how we have the capacity to engage and action that. Let's just reflect for a moment on who's actually doing any of this whole genome sequencing. Actually, Oliver has led the bioinformatics on a project that will be one of the first to be published on whole genome sequencing for a specific disease, and that's for Alzheimer's disease. And he's frowning at me, but I think it's true, Oliver. And it's been intriguing to watch that project happen with 1,500 or 1,600 sequences. But if you look at a world map, the big countries, the industrialized nations, except for South Africa and maybe the Brazil, is talking about or doing a whole genome project, a human genome project for hundreds or thousands or hundreds of thousands of people. There's going to be a 1 million sequencing project for America. The Faroe Islands is already well into doing 50,000 people. It's the tiniest nation doing it. And the English at 100,000, the Qataris at 300,000. These are large data sets, and they really bring to the fore issues around health and, of course, data sharing. One of the key aspects of that is the first thing people are hoping to find is ways to diagnose patients before they need diagnosis, to try and capture them from their code before they become an issue. And that's a very exciting idea to the NHS, and that's, I think, why the NHS has helped to invest 100,000 into the 100,000 Genome Project here in this country. I came here to this country to work because that was one of the factors that brought me here, the institutionalized structure around data. And as you know, working with data, not institutionalized as we should do with Neil, but institutionalized as instruction. I don't think that, Neil. It just came out. I thought you came because of the drizzle. The drizzle. When you did that, that's when my mind started to spin. So, talking of spinning minds, my sister and I share all kinds of weird aspects. She's more schizophrenic than I am, but certainly she has the disease very badly. And so I was incentivized to see if I could understand anything on her behalf with this company 23andMe, and that really is a probe-based analysis. So its assay is based upon the interpretation of an event where you have a predisposition, you bias the data sampling that you're doing on an array. It's not whole genome sequencing. And actually, it's quite interesting because I didn't really see much out of the results except a potential predisposition to bipolar disorder and an issue at the bottom which said potential developmental disorders in mental health, which was good, I thought, given that the integrity of that assay is questionable. And I read an article by Kira Peikoff, who is a freelance journalist working for the New York Times, and I've really enjoyed a dialogue around this concept that she brought up, which is, let's test the tests. So she went ahead and took DNA from herself via saliva and sent it off to several companies, some of which are on this slide, and asked the question, what does the diagnosis tell me in a consistent manner? And actually what came back is very intriguing. So at that time, there were 240 health conditions and traits that 23andMe would offer a view upon, and they'd give you the relative frequency of the alleles, or the pair, one of the two pairs that you have for each of the representations of a gene in your genome, and a nucleotide at that position is called an allele, and they take a look at that and say, what is the frequency that you have of this potentially deleterious allele or protective allele with respect to the population from which you're from. So they would population base you, so you're a Caucasian in my case. I'm 2.5% African, I'm very proud to say. I love the fact my ancestors were up to that kind of stuff, and that gives me some strength in this environment. 23andMe said 240 conditions. Other companies are more thorough, but have a lower breadth, 25 disease traits, 24 disease traits, and they asked the question, Kara, what's your potential to have psoriasis, which is a disease that I suffer from? And actually, one company said, which is 23andMe, she had a 20% chance of having an elevated risk for psoriasis, which is actually the same readout that I get when I take that test. And another company, she had a decreased risk by 2%. Okay, so this is the current state of the art in over-the-counter gambling about what you know about yourself. And it's because of the data acquisition bias, basically. What we're looking at is a situation where there's not a lot of information about the frequency and the sampling bias with respect to how you actually sample the assay. In other words, there are other contributing factors, or there are other biases in terms of the statistical interpretation based upon the sampling strategies you have will nullify or support your assay. So it's $99. Why should you care? Well, you should care because it's your data. And this is very interesting. So Illumina, which is the current monopoly sort of sequencing company, has the largest amount of name recognition, has created a new company called Helix. And Helix, intriguingly, is there to help you interpret your genome if you want to drop cash. And effectively, a subject will take saliva and spit into a bottle and send it off. And then you can't read this because I didn't realize we're going to have this cast. You'll get back a genetic profile like the one I've described in about a week, but it will be based on some aspect of sequencing of your genome. And then that will go into an applications ecosystem. And what happens there is that your genetic profile becomes one with an ecosystem which you can then pay to access contributor apps, which will then action your genome's analysis in some way, and there'll be some kind of ecosystem of interpretation. This is actually set up by Illumina, and they have an app ecosystem currently, which they're now commercializing to drive this concept. The apps are not best practice necessarily, but they're there. What is interesting, though, now is another company, HLI, has come to the fore and teamed up with a company which is Discovery Health, which is a South African company for its sins. We drink beer, and most of the beer you drink in the world today is South African beer, South African breweries. I love to say that to Americans when they come to South Africa. They say, I'd like a South African beer. I give them Miller Lite. And Health Insurer is another one, and there's a potential for 4 million patients in that system to pay $250 to a company called Human Longevity, Inc., and I'll talk about that in a moment. And for that $250, you'll get a whole exome scan. That means a less biased scan, because now the scan is not via probes that are sampling a genome at specific locations and imputing the rest of the data, but instead we're looking at actual sequencing of small segments of the genome. Anybody know what percentage of your genome is an exome? Anybody want to guess? 90%? 10, 8, 2, around there, right? So we're looking at a tiny percentage of your genome that's actually this part, but that tiny percent has a much better characterized function. Some of that, about 2/3 of that, is protein coding, and that allows us to start to infer function. And that's the first time I've mentioned the word function in this talk, but most of the talk will be focused on that. And the issue there is that's going to be a more predictive arena, because there's many exome data sets appearing, and already there are 90,000 data sets accumulated just for schizophrenia, and there's about 1 million exomes so far available for people to look at in the public domain. So it's becoming statistically robust enough for us to do sampling, and that's the basis behind that. But the concept really is that Craig Fenter, who invented express sequence tag sequencing, he likes to think about himself quite a lot, and he has a yacht. Apparently, there's a picture of his face on the sail. I'm just kidding. I know Craig, he's a funny guy. But he's offering this full DNA sequencing at the exome level to consumers for $250. And actually, it's a great idea if you want to give up yourself, because there is no guarantee that his company will fail and somebody will buy that company, or what will happen to that data no matter what happens, unless you're protected legally, which is why I was so intrigued by our discussion earlier. And actually, if you want to drop some more cash for $25,000, you can do an executive physical on steroids. And that means you can have an MRI, you can have an endoscopy, you can have whatever other place they can stick things, you can do everything. And you can also have your genome sequenced whole genome. That actually only costs about $1,000. But then the interpretation will be iterative. And the idea there is that after your omics makeover, you will be up on what may happen to you. And actually the incentive of that is getting back to our friend Craig, because Craig's genome snuck into the human genome sequence. And actually a lot of the joke in the community is that's a Fenta SNP, we say, because one of the nucleotides that we see came from his sequencing his own genome. Anyway, enough of the jokes, but basically it gets to the point that the viability of that model is that the company is the database. It's the data that matters, that database that you're giving up. They're not making money selling you kits. That's the freebie, that's the mobile phone. They're getting you on the data plan. And effectively, the data plan is a good idea, but to do it commercially, I think, is a challenge because of this issue of checks and balances. But then the question becomes, given what we've seen in terms of Oliver's work and others, capturing human variation at the level of disease is enormously important. And capturing that at the level of the code that codes in part or fully for the onset of that disease is, of course, enormously important. The problem is it's still new stuff. So guidelines for reporting disease-causing variants is still very much an area of flux. The American health authorities have a guideline set. They have a club that they put out their ideas. The British have a club. They put out their ideas. Everybody has ideas on what those reporting standards should be, but that affects effectively what gets back to you as a patient. And the evidence for candidate genes is very much an area of flux as well, because even if a nucleotide changes and you see it in the genome, that doesn't tell you that that is causal. There are a number of things you sort of necessary and sufficient that you have to do, and unfortunately, you can't do that much experimentation on humans. So it becomes a challenge, and this challenge of next generation clinical diagnostic sequencing is heavy. There's a great paper by Dan MacArthur on it in in Nature this year, and it's worth taking a look if you're interested in this area. The British have teamed up with a Swiss company to take a look at a pipeline approach to this problem. You can't see it very well, but basically you go from clinical next-gen sequencing where you look at lab validation, and that basically means a bunch of people reading your genome sequence and then reading papers about it. That's how sophisticated and scalable we are right now, and I don't think it's going to change very fast. It's actually very slow. Then you go through the system where you get to the concept of clinical genomics, where it's possible to have a pop-up screen which says, do not give this patient warfarin. They won't stop bleeding if you do, and that can be based on a genetic test. And then you go through to clinical reports, and hopefully to the point of some kind of clinical enlightenment. It doesn't really exist in the UK yet, but it will, presumably. But certain universities, such as Vanderbilt Hospital and University, they're certainly driving this particular concept. And this is something which the American medical informatics community is quite excited about. So interpreting these single nucleotide variations that you see in the genome, some of you do that for a living, I'm sure, if you're working in statistics. It is a very challenging arena for some of the reasons I've described. One of the big things that comes from that is that you might see a trait that be continuous, like height, or discrete, like pregnant, and basically those traits can then be associated with your genome code. And if you have enough samples, and I mean enough samples, you can perform a statistically robust test which allows you to say that this elevation in frequency of this particular allele is representative of an association above a statistical threshold for that trait. That genome-wide association then becomes intriguing. But the majority of the traits of the signals that have been seen to date are not coding. They don't sit in this exome scan that I've just described. They sit out there in space, and we're not quite sure what their relationship is because we don't have the genome features of the rest of the genome well characterized. The other problem is that when we're trying to figure out this interpretation of whether an association is important or not, is that there are 2 alleles, so it's a frequency. And the other problem is that the genome is not 2-dimensional, it's 3-dimensional. So the relationships between a variation here and another variation here, or a functional feature here, which can be on the same chromosome, on a different chromosome, or on a DNA interacting piece of protein that's sitting on the inside of the nucleus, have to be determined. And technology called Hi-C has come to the fore that allows us now to look at that structure. And intriguingly, the 3-dimensional structure of the genome has aspects of chaotic organization. So it's quite intriguing that you can apply mathematical principles to the structural features. So the other aspect of this is kind of where I look at this work is functional interactions. And that is what happens once you've interpreted the code off the genome and have transmitted that information into the cell or into the system. And that functional interaction occurs after transcription and translation. And that means that you're sitting in a 3-dimensional space where you're having interactions with the environment or the internal environment of the cell, and that's responsible for the cell to go through an expression state or a time instant state of what's occurring. So you're beginning to see how multidimensional this problem. I was talking to a graduate student earlier who was talking about a study that she's working on in the reduction in multidimensionality. This is the mother of all those problems because there's just too many dimensions for life, but effectively it's an orchestra. So getting onto orchestras, a colleague of ours, Alban Sanderlin, is looking at promoter regulation, and that's the region of the genome which of course is upstream of each gene, which helps us to regulate the expression of that gene. But actually there's many regions which do that, many kinds of regulatory influences at many levels, and that regulatory interaction we call an orchestra, and determining how variants at critical positions define what kind of song you play is the art, effectively, that we're getting to at this point. So one of the ways to get down to that interplay between off-genome events, expression, protein translation, translation to protein, protein interaction, is to look at variants in the genome that vary, that are seen to associate with particular expression states. In other words, you get a particular expression profile of the transcriptome, and that expression quantitative trait loci, something which is pioneered in plants, can then be applied to this information if you have state-specific gene expression information. I spent a lot of my career looking at state-specific gene expression information, so once again, picking that previous ability off a tree, it's effectively the same as you are in your developing methods, and then translating that into applying that now to what we have as a new problem, we can start to understand the trait distribution and look at the SMP distribution. And finally, you can ignore all of that and just say, what genetics does a person have? What allele do they have at a particular position, at a particular frequency? Can we stratify the population based upon that? That's a simple idea, and the statistics in that is becoming more and more robust, and that's basically where the majority of the work is being done right now in patient stratification. The point being that if you can stratify a population and stratify it according to how they'll respond to a drug, then you're likely to be able to treat that patient more effectively. The problem with that, incidentally, I was very excited when I first learned about how this works. I actually went to doctors in Yorkshire, and I met a few of them, and I asked them, wouldn't it be better if you were able to do better at telling which patients are going to respond to the drugs that you treat? And the answer was shocking to me. The answer was no. I don't want to know that I'm going to give a drug to a patient that isn't going to work. That patient doesn't want to know if it's going to work or not. I want to give them hope. I don't want to give them drugs that don't work. But at the same time, I want to give them drugs that do work, obviously, but if in the majority of the cases it's not going to work, I still want to give them the drug so that it gives them hope and it gives them some form of quality of life. And that is the prevailing attitude amongst general practitioners that I've discovered in Yorkshire. We have a long way to go in terms of utilizing this information effectively so that it actually is useful. You may know of Terry Pratchett. He has a particular locus, ABCA7, and he died of Alzheimer's, as you may know. And that locus, if you have it, has a predisposition to early onset Alzheimer's. This is what he died from. And they didn't know that when they first started to interpret his genome. First started to interpret his clinical presentation. Now, of course, it might have changed the way he was treated in an earlier stage. But what we're looking at now are new variants which can give us information about onset of Alzheimer's. And that information is useful to us, of course, because if we have that information early, we can help people before that dementia sets in. another famous person who has motor neuron disease, which is the focus of work that's performed at the Institute, the Citran, where Neil and I both work, has a progressive weakness of his musculature because he has a genetic predisposition that we don't understand. We just don't know what's actually giving him that slow progression. Motor neuron disease usually has a very short lifetime before you pass away, and the penetrance of certain mutations, some of them as high as 10% in people who have motor neuron disease, can be very predictive of outcome. So scaling up, the only way to capture all of these rare variants that give us insight into how a person's going to progress into disease or whether they're going to get the disease is to sequence a scientifically termed shitload of people. And that allows us to get an understanding then of the spectrum of variation that contributes to the disease. And what comes out of that is that we often find it's not just a single gene giving you the disease, it's a concert of genes interacting in some way, or promoter elements or other aspects of the genome with the environment, in this case, physical stress. So you're getting a sense then, I think if you haven't been in this area already, of the enormity of the dimensionality of the problem and the enormity of the challenge that we face in terms of interpreting that. That doesn't stop people. I think one of the best things that's starting to happen is that there's a move away from model organisms. As we heard earlier about model organisms this morning, but there's a move away from using model organisms to test drugs. What we're doing now is preclinical validation of drugs in humans who are prepared to take that type of test. And I always think of Charlie in the movie, in the show 2.5 Men, who had no money, and he went and took a drug and he developed a lot of pustules and weakness and stuff, and it's kind of funny. But actually I think it's an effective way, and drug companies are very keen to work with potential treatments by doing this. So we go away from veterinary therapy, in other words, fixing mice, to fixing humans. And one of the ways also that there's a potential is we heard earlier about this idea of induced pluripotent stem cells that become pluripotent because they've been they become transdifferentiatable by virtue of giving them a cocktail of transcription factors which changes their landscape of development. The problem with that, based upon static epigenetics, which they're captured before they went through the transdifferentiation process, is that they may or may not represent good models of the disease, and they're in a dish. So you have an in vitro model from a human that might be a patient, but the response to the treatment might not be indicative of the response of the patient. So that translation is something that really interested me while working at the Harvard Stem Cell Institute together with Oliver and others. And effectively, this idea of transdifferentiated cell models was pioneered there in terms of treatment. And Clifford Wolff helped with this particular work to show, together with Kevin Egan, that you could discover from a genome a mutation, develop in a induced pluripotent stem cell model a model of that, find out that the excitatory behavior of the motor neuron that you've transdifferentiated is different, and predict a drug that might be able to treat that particular transdifferentiated model. And they did that, and they showed they could improve the excitatory ability or change the excitatory ability of a motor neuron as a result of the fact that it had this particular mutation. Now that holds out hope for the patients. The drug they used is one which doesn't work well in humans, full humans, but this shows you that they could show a good proof of concept. I was very excited by that. The other aspect of this transdifferentiation model is regeneration, and Doug Melton, both of his children have type 1 diabetes. It's type 1, isn't it? Or is it type 2? I think it's type 1. He's very incentivized to help them with that, and effectively he's built a stem cell-derived pseudopancreas, which now allows potentially mice to recover from type 1 diabetes. And obviously the move forward to clinical trials is very important. That's a transdifferentiation problem, but actually it's a systems biology problem because you have to understand all of the interacting factors moving together, the moving parts together with that cell. So getting onto the systems biology approach, we've talked about variations in the genome, and we've seen that you can interpret a model based upon the fact that you can induce it in a human system. But then there's this issue of treatment. And one of the ways to go there is to look and break down the multidimensional approaches. Here I'm looking at fly, mouse, human, and cohorts. So we're looking at these structures, which are representations of models in different systems. Then we can use genetics, iPSC models, genome variants, etc. We can look at expression pathways, expression QTLs, omics, and with each iterative application to a network model of the disease, which can be made up in this instance of genes and their connectiveness based upon their interaction or their co-expression, you can build models which become more and more selective as you improve the amount of information. It has to be of good quality, and we heard a lot about that earlier this morning from Oliver. You have to reduce the noise, and you have to improve the signal, but as a result, you can narrow down on a module of interacting genes that seems to be very relevant to the disease. And that's really where the state of the art is today. How you narrow down to that, there's many ways to do this. These are ones of thousands of approaches. But effectively, that narrowing down is both algorithmic and data-driven. And it's very much a case of having good data. A recently published model for Alzheimer's puts a couple of diseases, it puts a couple of genes in the middle, TYROBP being one of them, derived computationally and then established through mouse models to be effective. But the treatment, there's no treatment for Alzheimer's. There's been no resulting translation. So let's go there. How do you translate? There was an earlier question we had, and I wanted to answer in Greek because it's all Greek to me. I'm sorry. This is it. It's a captive audience. I can't help myself. Preclinical validation, standard and elevated do the same thing, right? So the preclinical validation is actually the state of the art right now. And I'm very excited to be part of a consortium that works between industry, between geeks like me and maybe some of you, and the data generators, the people generating information from people who have passed away with Alzheimer's and their brains have been selected out, and normal people who have given up their brains and doing comparative case control studies. And effectively what we've ended up having are network sharing consortia. And the Advanced Medicines Partnership, which is driven by the National Institute of Health in the United States, has built one of these network sharing consortia around Alzheimer's disease. I was very excited. I listened in on a call yesterday. It's making a lot of progress. But it's a preclinical consortium. And that's the concept. You have companies who pay to have data generated and have it analyzed so that they can get a model for the disease. And then what they do is they even openly validate. Takeda Pharmaceuticals is an instance of that. And they say, give us a way to help us rank our targets. Let's do some preclinical validation in the public domain. And then they go off screen and use their drugs and do their own story. So they're working more and more, in this instance, in the public domain. It's a risk reduction exercise. It's a crowdsourcing exercise in a controlled fashion. And what's intriguing about that, actually, I heard yesterday about the exact method that SmithKline is using to rank targets. Our own institute has now created something, I've just created something called the Center for Genome Translation, where we're looking particularly at discovery of ranking of targets for, those can be processes or genes. And then downstream of that, there's another institute at the Sanger EBI, which is called the CTTV, and I don't quite know what that stands for, but it's got the word target validation in there. So we're doing discovery and target validation in the public domain, giving away the data, but making it possible, and all of this data incidentally is available through this consortium, and making it possible, if publicly it's not, there's no strings, making this process of preclinical targeting much lower in terms of risk. So my view is that in order for us to action the tremendous amount of data we have to reduce the dimensionality, and we have to understand function a whole lot better, because by doing that, we can quickly move to target prioritization and target generation. So to that end, we developed something which allowed us to compare functions quantitatively. And that's never really been done. There's a lot of that done in terms of gene set enrichment, but very little done in other ways. And Oliver here is one of the major contributors to that concept, but a group of fantastic people I was fortunate enough to work with recently at Harvard helped us develop this method, and it's been led by my ex-student, Gabriel Altschuler. So I want to go over the method with you because it's very simple, but after that I'll show you how we're applying it in this translational space. So what we do is we take a gene expression experiment and we rank the expression of the genes in that experiment, and then we take any gene set, if hopefully one that has a known function, say the Wnt pathway or MAP kinase signaling, something that has a known entity, or we take something which has been data-driven in terms of its production by a group of interacting or co-expressed genes, and we ask what is the rank in this gene set of the substituent genes in that pathway, and we just take the rank statistic, and we take a score, we sum the results and put it through some manipulations, and then take that score for that ranked pathway and pass it across gene expression sets from all kinds of different organisms, including human. So we take, obviously, homologous genes when we're looking across different organisms. And we ask the question, do we get a distribution? And we do get a distribution for the activity of that particular pathway in that system. And the extremes of that pathway activity, we then rank into tertiary scoring, 1, 0, 0, -1. And that ends up allowing us to have a signal for each of the elements in that particular pathway. And we just take cutoffs from the extremes of the distribution. The result of that is that we're able to build a vector of pathways or gene sets, and we can build that for each organism. We can build that for a particular expression state, a particular sample. That means that we can fingerprint at a functional level the activity of a single sample in the context of all other samples in excess of 100,000 that we've looked at. What we are then able to do is ask, if we look at a particular pathway in that fingerprint, is it on or off, or downregulated or upregulated? And we can say with respect to the distribution, how is that? We can also take any two samples and ask that same question. And that's actually quite a powerful endeavor, because it allows us to put that particular sample into a distribution for a disease. And you can see if you have patients and each one of them has a sample of some sort, you can get a distribution of patient activities, and that's something we do right now. We've done a study recently in multiple sclerosis to investigate that. And with that pathway fingerprint, we can find that there are many applications of this concept in disease. We can do survival outcome, and I noticed that there's some work on that here in this group. And we've looked at improving survival in endotoxin sepsis using that particular methodology. I'm giving you no data because I don't have the slide. And basically, we're also able to map information from the genome and genome variants. This is actually a little bit of work that Oliver and I have experimented with, where effectively you take a genome-wide association study and you take variants which are associated with some kind of condition, and you see if you find similar outcomes in expression space. So you're moving between two different incoherent data sets, one of which has sampled many, many hundreds of people, another which has sampled many, many thousands of experiments. So you have a relatively statistically robust background, but then the question becomes, can you use that information? So I've mentioned this concept of the GWAS, and if you look at each chromosome using a Manhattan plot, you can land the helicopter here, or you can land the helicopter here. And basically, most of the studies say, let's land the helicopter here, but what's happening down here in the noise? And the question is, these are variants occurring at specific frequencies on particular chromosomes as you go across a particular trait. And the question is, can you link that in some way to function? And these are some slides which I tried to dig up that Oliver made years ago, so forgive me, Oliver, for digging them up and not asking you about it. But effectively, you can find that certain single nucleotide, what does P stand for? Polymorphisms. I want to say single nucleotide particles for a second, excuse me. Anyway, so thank you. So the polymorphisms can effectively be used, and they're not necessarily going to lie within a gene, but they may be associated with a gene. If you link them to a gene name, then you've got the ability to say, here's a gene name which I can plug in to this Pathprint system. That allows us then to draw out pathways which appear to be associated with a particular disease from a genome-wide association study. And here are some pathways for a particular lung disease. And we can ask then, do we see any relationship with that particular fingerprint when we look across all of our data set, and then do we find anything which is near it? So we go from genome-wide association study to gene expression study. And actually, you can find coincidence in quite a number of those pathways, and I think there's a number here like 9 or 5 or whatever. And when you look at them, they can be from samples which are very similar to lung disease. So lung adenocarcinoma is an example here. So that conceptually is how far we got with that particular analysis. But it gives you an understanding that by dimensionality reduction, you can then go ahead and ask questions of whether that's statistically viable. Can we see whether it segregates an experiment based upon survival? And effectively, it can be quite interesting. But to move from that pilot concept into translation is really a big, big challenge. And to do that, you need more data. So you have to be able to integrate across many of these dimensional modalities, and that means that you have to be able to have a methodology to do that, and you have to have infrastructure. This is a slow and painful process. And in science, as in medicine, we're always looking for the quick fix. So, this is one of the reasons I talk to communities such as yours or ours, and that is so that we can combine our approaches and come up with more effective models. I'm very excited to work with a colleague of mine who's been doing work of this nature, Yared, in my group at Harvard, and effectively he's been interested in the concept of internal pathway correlation or coherence. And effectively that means to what extent, if you look at two genes as they go across this sample of many, many gene expression experiments, do they cohere in terms of their coordinate co-expression? Sorry, in terms of the correlation. And how correlated are external genes with that coordination as you pass across this gene set-based coherence approach? And using that concept, he built a network not of genes, but of pathways and how they were interacting. We're doing all right for time? Yeah, okay. So effectively, what we did then is something just for laughs, as they say in Sheffield. I enjoy that very much. And effectively, we took a whole bunch of different pathways and we asked, to what extent do pathways interact, and can we cluster that interaction? And we just did a set of clusters, and we basically built a set of modules of interacting pathways across the cellular system. And what intrigues me is what came out of that, and it doesn't really matter about what the names are, but effectively we found that there are 4 basic clustered functions in the cell if you do this quantitative approach. One of them is signaling, cell signaling. Another is protein trafficking and degradation. Another is mRNA biology or mRNA world. And the final one is immune response. And those are the 4 biggies. So what intrigues us is that by taking that structure, you can start to annotate that structure with disease and start to see how parts of the disease work at the macro scale in the cell in interacting with each other, built upon a static model, but giving you the ability to tweak that model with respect to each disease state. It also allows you to take well-known systems which are built up into gene sets, such as the gene ontology, and refactor that based upon a data-driven approach as opposed to a domain specialization approach. And if you put in axonogenesis as an example, these are the results you get back. The stuff in red is now no longer readable. These are downregulated processes. And you'll see that cell cycle is downregulated for some reason, or is negatively associated with axonogenesis, but transmission of nerve impulses is positively associated with axonogenesis. Interpretation of this is almost impossible because we're looking across many domain sets and we have to use something statistically robust and we have to validate it. And I think one of the things which Oliver was also mentioning earlier on is you want to work in an environment where you can come up with an idea, work with collaborators that enjoy what you're doing, you enjoy what they're doing, and move the whole way through to find the data-driven results, but then go in and show that what you're saying is both necessary and sufficient to cause that particular event. This is a slide on the right, stolen from some of Oliver's co-workers in Scotland, and sorry, it's a slide was stolen. No, it slipped out, Oliver. So, sorry, I'm teasing him because he went to Scotland. He didn't come to Sheffield. And why would you do that? But the issue is that I stole the slide from him, but not the content. This really gets back then into stuff which is not exciting necessarily to somebody who wants to develop algorithms, but is absolutely essential if you want to translate this work. You have to have provenance for your data, reproducibility, you have to have established data sharing paradigms, and you need to think about precompetitive approaches. These are keys to translation in this modern data arena. One of the big things which was commented on recently by Lincoln Stein, another of our friends who works in Toronto, another very proficient computational biologist, is that we really have to think about ways of sharing data appropriately, and his push has been to cloud. And effectively, the idea is to bring genome data and genome analysis together with cloud, and that cloud approach, together with concentrating algorithms around the data as opposed to data around the algorithms, is really how we have to go. That's challenging because that means refactoring algorithms and thinking about new ways to work. But it is the way that preclinical studies are very active. And in fact, I don't know of any preclinical large-scale analyses currently being performed by consortia that are not using some aspect of cloud interaction. 2 examples of that: Sage Bionetworks, based in the US, has really got into the idea of nano-publication of data workflows, which allows you to bring your studies together into one web accessible and web service accessible space, and then to work to publish the analysis you're doing with a document identifier, so that not only is it yours, and people can find out who did it, but it's also got provenance, reproducibility, and distributability. We've adopted some of those concepts in our own Harvard Stem Cell Commons, which is also in this arena, where we publish data sets that have been put into data publications. And I'm very proud to say that Sheffield University is thinking hard about this concept of publishing data internally and publishing it externally through an interface that allows us to work with the open access consortia. Why is that interesting? Well, the work that you're doing needs to be publishable. And the levels of publication are not binary. There's actually a decline or a distribution of those publications. And I think Neil's a great example of that. You know, a lot of what you think is on the web. I mean, just it happens. And I think that really is very important in our field to interact with our community and to draw new people into our community at many, many different levels in order to do iterative peer review along the whole process. So I'll skip that slide and get to this point. And this is basically one of the last slides I wanted to show, which is that in the concept of building a center for genome translation, you have to work in so many domains. You have to work in the disease domain, where you're working with clinicians and you're working with biorepositories, and you're working with sample data and metadata, and those have to be described very, very well with well-structured phenotypes. And then you go through with that phenotype information and allow that to be compared with disease models that you've built. And those disease models, as you know, can be in silico, in vivo, in vitro. And the idea there is then to have information from genes and to build a continuous iterative model. And the key, once again, to success in this process is not to stop. You have to keep iterating to improve the value of the model. The key right now, the sort of state of the art, is very, very highly communicating groups of interdisciplinary researchers. That is the key to success in this arena. So what are we looking at in the future? A few ideas. The data patient, real-time trials. So as people are taking the drug, they're being monitored and quantified to find out how they're responding the whole way through the day. There's no visit. There's no visit. They use an app. They get everything in the post. They're consented via the app, and the app is watching them. And that really is, I think, one of the things which is really starting to happen. There are already 20,000 apps in the App Store that are health-related, and a small handful of those right now are working in this particular space, but it's growing very rapidly. I've seen exciting new studies in Parkinson's, for instance, that are using that approach. The predicted patient, keep the patient out of the hospital, using some of the techniques I've shown you today and others, particularly about tracking that patient and their behaviors. Let them know how to change their behaviors. And actually there's an app which my colleague Eric Schatt at Mount Sinai has termed the Death app. And that app helps you understand your heart circulatory associated behaviors. And it's in the App Store. I don't think it's called the Death app, but he calls it that. And what it does is it's monitoring your heartbeat and all these sorts of things using your Apple Watch, etc. And it's saying to you, given the way you're behaving right now and what you've just eaten for this week or today, you have a 0.05% chance of being dead by the age of 30. But what it does then is it comes back and it says, your chance based upon your behavior last night is that it's now 0.3%, and it goes up. And what happens is as you get nearer and nearer to misbehaviors, it'll say you must get to the hospital within the next 10 minutes, otherwise you're toast. And I think that's really the idea, is that predictive, keep you out of the hospital approach is very financially viable. And the UK government has started to clear up on this as well, which I really like about the approaches here. It's just spent £20 million as a result of re-electing the Tories and their election promises on connected health, which is what it's called in the UK, to build systems which allow us to do predictive work. There are other aspects of this in machine learning, and you do well to take a look at the hodgepodge of approaches that have been put together that's been termed Watson. And the IBM Watson project is to ask the right questions based upon training, given the answer. And that was how it won this American show Jeopardy. It's now employed at a number of places as a product, a commercial product, that allows some form of predictive behavior based upon omic information. It's still very early days, but the translation of that directly into clinical records is occurring as part of that project. The one showstopper for this is that it's one of the keys of IBM's future trading philosophy, business philosophy, and as a result of that, it's it's priced out of the market right now. The interaction between genetics, and the environment is a very important thing, and quantifying the environment and quantifying genetics successfully in a coherent manner, in other words, where you've got the subjects and the environment down, is the wave of new data sets for the future of looking at precision medicine. And finally, I love this concept of face space. And that is, first of all, you can predict how people look based upon markers from the genome. And there's art to that effect that's published in New York from cigarettes that people have discarded and picked up, and then they've modeled it and so showing that it was probably an Iranian guy with a beard, whatever, that was smoking that, all the way through to forensic technology. And at this point, you can produce recognizable pictures of yourself from your DNA that you've never seen, and people will be able to pick you out from a timeline, a lineup based electronically on your face as opposed to from a prediction. I'm worried about social networks and genetics, and I'm sure we'll get to the point at which aspects of our genomes will be shareable by ourselves, and we'll be able to find interesting people based upon that, and that opens up a whole new kettle of fish. But I really think it's possible because it's known that individuals who are friendly with each other are more likely to share aspects of their DNA than that individuals are not, above some cutoff. So we're on a wonderful new journey, and I'd like you to join with me in thanking the individuals that have helped us to build this study, in particular Oliver Hoffman, who's with us today, Shannon Hosui, who's a colleague of mine at Harvard, whose birthday it is today, Khaled, and my group in Sheffield, particularly Gabriel Altschüler, who's done a lot of the work that you've seen this afternoon. Thank you for your time.